We are gonna start with the 3 minutes. 3 minutes thoughts for this session. I promise I'm gonna cover different types of organisms, and our next talk is by Ralf Sommer, who is a director here in Stanford Biology, telling a story about nematodes.
Okay, thank you, Lisa. So my lab studies a phenomenon that has been touched upon already a few times during this meeting, and that is developmental or phenotypic plasticity. Our study system contains a strong element of noise or stochasticity, and it turns out that it is actually this element that makes this particular example of plasticity attractable to study and to learn about the underlying genetic but also non-genetic processes associated with it. But before I get into this, this is not working, so I do it this way. Ah. There we are. Now it works. Okay, then I'll use this. Yeah. Okay. So because given the fact that plasticity is a contentious term, it is important to really define what you mean with it. And in our lab, in our context, we consider developmental plasticity the phenomenon where a single genotype produces multiple phenotypes in response to variations in the environment. This phenomenon is omnipresent in plants. Zoologists have been resistant to for a relatively long time, although you see from these beautiful pictures that also in zoology you find plasticity really all over the place. But the convincing argument that it is important came only with a monumentous monograph of Mary Jane West Eberhard a little bit more than 20 years ago, where she showed once and forever that plasticity is pervasive in nature. It is a major neglected factor in evolutionary biology, and at the same time, she proposed that it is a major facilitator of evolutionary divergence and novelty. I threw in this slide for Luisa to point out that really plasticity can come in many different flavors, and that has presumably contributed to the confusion. So plasticity can be reversible or irreversible very often in a physiological context. It can be continuous or discrete, the latter being very often referred to as polyphenisms. And here it is important that in the field we believe that these examples of polyphenisms are secondarily derived, where you throw in a threshold, and if you are above a certain threshold, you have the one phenotype. If you're below, you have a second alternative phenotype, which is then different from the example of continuous plasticity. Third point, plasticity can be continuous or at least in part stochastic, and I will return to this. And of course, from an evolutionary perspective, very important. It can be adaptive or non-adaptive, and for those that are not so close to the field, there are surprisingly few examples where people and study systems could show that plasticity is really associated with and has an associated adaptive value. So there are a few study systems out there, and that has also been contributing to the reservations among evolutionary biologists. So we do not really know at least until 10 years ago, what are the associated molecular mechanisms. And yeah, question then arises, what has been the problem with studying the role of plasticity in evolution? And for that, we have to return to this picture of these beautiful animals, but something that they nearly all share is none of them is a lab rat. None of them can be easily studied in the laboratory, and therefore it was very, very complicated to get an understanding on the associated mechanisms. That is where nematodes come in. I guess you are all familiar with Caenorhabditis elegans and its multiple advantages as a study system. In my lab, we have developed more than 20 years ago this nematode Pristionchus pacificus as a model system for evolutionary comparisons to C. elegans and for Evo-Devo in general. I'm very brief on this. Pristionchus belongs to a different family, is separated from C. elegans for more than 100, presumably 200 million years, but nonetheless, it shares with C. elegans a number of important technical features. It's again a self-fertilizing hermaphrodite, very short generation time, can be grown on single bacteria as food source, and that allowed us to develop more or less all of the tools that we have available from Caenorhabditis elegans as well. The advantage when it comes to plasticity is that Pristionchus comes in two alternative mouth forms. During post-embryonic development, each individual has to decide if it wants to form as an adult 2 teeth the so-called eurystomatous form, or a single tooth, the so-called stenostomatous form. After that decision has been made, it is an irreversible decision, so they cannot go back and switch to the other form. So it's a very important decision, and it's not only has morphological consequences, but also behavioral consequences, because it's only this eurystomatous form with 2 teeth that is a potential predator. They can still feed on bacteria, but they can also predate and kill other nematodes, whereas the stenostomatous form is a strict bacterial feeder. All of this happens in a self-fertilizing hermaphrodite, so genetically, all of these animals are absolutely identical. I just throw in one very brief slide that we, of course, have to look at this in the context of the ecology of the organism. Pristionchus can be found in the soil, but it is most reliably found in association with scarab beetles. These scarab beetles fly around as adults. The nematode sits in the folds of the cuticle in an arrested dour state, waits until the insect finally dies. This happens in the soil, and then the nematode smells the growing bacteria and fungi on the carcass of the insect, gets out of the dour stage, and then the clock starts to run, because the food that is available on the carcass will only be there for a certain number of weeks, and then it runs out of it. And therefore, certain nematodes, like our Pristionchus, have played the game in being able, in addition to feed on bacteria and other microbes, also to be able to predate on other nematodes. So what is very important is that our system of plasticity has to be considered as a bistable epigenetic switch, very different from what we see in seasonal polyphenism in insect or butterfly wing patterning, and more similar to what you know from bacteriophage lambda. There is an element of noise or stochasticity in that even under fixed environmental conditions, each strain will still form both mouse forms. This here is our Californian wild type. If you grow that under standard nematode growth condition, E. coli as food, you will see that you get in a certain ratio both mouse forms. And when we saw that first time 15, 18 years ago, we thought, wow, that means presumably that we can very easily manipulate the system. And that is what we can indeed do. We can create any ratio that you can imagine. We can genetically manipulate it, have isolated mutants where this one form is no longer existent and they are all of the non-predatory form. We can play around with the environment. We stay with E. coli as food, but go from agar plate to liquid culture, and you see we can reverse the phenotype. We have by now more than 1,500 wild isolates from around the world, and you can see on an E. coli diet, they can come in any different phenotypic range that you can imagine. And if we then even start to play around with the diet, changing away from E. coli, then again, you can see that we can very easily manipulate the system. So therefore, we have here, under any given condition, a probability of forming both mouse forms, and that is strain and environment specific, and that is very different from the situation that we see in butterflies and other systems. So these somewhat unique advantages, I would say, of the Pristionchus species: the binary morphological readout, no intermediates, the fact that the hermaphroditism has removed all genetic polymorphisms, in the very easy way of genetic manipulation, has allowed us to aim for a fundamental understanding of developmental plasticity as a facilitator of phenotypic evolution. And to achieve this, we have been asking 3 questions over the last 10 to 15 years. First of all, how does it work mechanistically? Is it of ecological relevance? And is it significant for evolution? And out of these 3 questions, we have derived 3 major research directions. And at least the first one, the aim to understand the molecular mechanisms of its regulation, we are pursuing for more than a decade by now. So how does it work? We have been able to show that there is a very detailed gene regulatory network of mouse form plasticity, including many genes. I will not dare you with any of the details. I just say that at the center of this are sulfation processes. So sulfatase has been identified that if you eliminate this, this is a major developmental switch, and one of the 2 mouse forms cannot be formed. At the same time, there is a sulfotransferase that does just the opposite effect. And if you knock out this gene, you have only the opposite phenotype. So you see there are developmental switches that really sense environmental information and will then run the system in one direction or the other. We have also, because that has been a discussion earlier on today, we and others have isolated mutants in epigenetic modifiers that can really change the standard ratio that you have in a certain strain into 50-50, from coming from 80-20 ratio in one direction into 50-50, or even into a majority of 20-80 now in the other direction. So we see that in addition to these genes that control the process per se, there are also genes that control the stochasticity and the element of noise that is in the system. But the most important take-home message from this slide here is evolutionary biologists don't have to be afraid about plasticity as a phenomenon. There's nothing mysterious behind it. These type of processes are regulated with the same type of logic than other developmental processes that are canalized and that are not plastic at all. Is it of ecological relevance? For that, we have been going into the field for a long time, but a couple of years ago, we have started to do agent-based modeling approaches. This is work of Atta Kalirat, who is here in the audience, and who has a poster tomorrow morning. So I will encourage you to go to his poster, and I will leave this up to the discussions with him. And therefore, I move on to the last question, which we have started more recently. And that is the question, is it of significance for evolution? And in order to get into this, we have started long-term environmental induction experiments. This work was started at the beginning of the pandemic, where we couldn't run away. And this has been done by, and now the Tübingen graduate students close their ears, not by an outstanding graduate student, but by a unique graduate student. And so what she has performed and is still performing is just amazing. So what has Sheila done? Sheila has taken a strain from our field station on Reunion Island that is preferentially non-predatory. She has taken a single mother and singled out her complete brood. So all of these animals, to start the experiment, were sisters and genetically identical, 110 of them. And she has not grown them on the standard E. coli food, but on another bacterium that was derived from a Pristionchus environment. And we knew that this food would enhance predatory traits in our wild type strain from California. And then what she has done is she has done single worm transfers for 100 generations of all of these 110 lines to see what will really happen to the system. And the idea here was, we know there is no genetic variation in the system, but we wanted to capture epigenetic stochastic effects in the system. And for that, we have always transferred only a single worm to enhance the chance that such an epigenetic event had a chance to manifest itself. And then in a certain generation, in generation 50, 25, and so on, we have started to revert all of these lines also back to E. coli to see if something had already changed, or is there a memory or whatever. So I summarized this very briefly. She found three major patterns. First, what she found, and you see now in all of these graphs, generations on the x-axis, percentage of predatory phenotype on the y-axis. What she found by switching to this novel bacterial food source, dietary induction of the predatory morph. All of the lines, all of the control lines here remained 20 to 30% predatory as they were before. The moment you switch them to this new bacterium, they are all 100% predatory. This is immediate, this is systemic, this is permanent, and they would never go back if they are kept on this diet. The second phenomenon she observed when she started to revert them back to Escherichia coli. Here is the last generation on the novel food source, novel syngrobium. Then you revert them back, and you see these guys keep a memory for the first 4 generations. They are still preferentially predatory, and only then they go down. And the longer you have kept them on the new diet, the more memory they will show, and the longer they would keep in this novel induced trait. And then finally, and that will be in the long run the most important finding, we found trait canalization in that certain lines with low frequency, usually 1 out of 100, when the others were all down, would not go back at all. So this trait can be canalized, And we saw this in every reversal experiment that we have done. 1 out of 100, but it was always a different line in each experiment, indicating that during the reversal, there is something, an epigenetic phenomenon that will finally manifest itself. And we have by now good evidence that these phenotypes that we see here are genetic and not permanent epimutations. So we are now in the process that we want to really understand what is the nature of the inducing agent of the bacteria, what is the mechanism of transgenerational inheritance, and all of these phenomena. And I will just throw in one final slide before I come to the conclusion, saying that we were able to tie up this long-term environmental induction experiment, as I described it before, with an EMS mutagenesis, because we wanted to know, is there a genetic basis for memory? And when we did this, surprisingly, this was still in Corona times, we could show there is a genetic basis for this. I will not bore you with any details, but we were able to show that small RNA, microRNA signaling is absolutely crucial for this phenomenon. And there's an E3 ubiquitin ligase that is absolutely crucial in the system. And the process we have by now been able to show involves target-directed microRNA degradation. So it's very sophisticated epigenetic processes are involved in this phenomenon. And we have already started to build models how all of this works. So the take home, we have a model system trained for studying molecular mechanisms underlying phenotypic plasticity. We observe diet-induced plasticity that is subject to transgenerational inheritance and trait canalization. These phenomena require microRNA signaling, and we are currently involved in understanding what are the triggers and targets that really work on this microRNA signaling. So very important, all of these epigenetic processes evolve super rapidly. If we do natural variation studies within our species, we see that there are major differences of these microRNAs. There are major differences with respect to memory. So all of this is evolving at a super rapid timescale. But we are therefore of the opinion that developmental plasticity really has to be considered as an evolutionary concept. And we usually do this, not only we, but also others, by assuming a 4-step process. You have a monotypic canalized trait that comes under the influence of an environmental cue. This will result in plasticity like we see it in our system. The moment you have this plastic trait, natural selection can act on both of 3 of these traits that you have. This is a phenomenon that several people have called genetic accommodation. And then sooner or later, we come to Waddingtonian evolution because the environmental influence might become genetically encoded. And that would then result in the canalization like we have already seen it in the lab. And in the phylogeny of our Pristionchus family, you see here Sinuophytes elegans as an outgroup. We see exactly all of these 4 steps. So it's really reassuring, not only us, people working with insects, people working with vertebrates have been able to show plasticity is very important. In our case, when we look in our family to which Pristionchus belongs, when we look at these teeth, then we see the teeth have evolved at the base of that family. And the moment the teeth were there, they were developmentally clustered. And that is exactly what West Eberhard has predicted more than 20 years ago. This is a predictor of novelty. You create novel traits by introducing phenotypic plasticity. The moment you have it, natural selection plays around with it like crazy. We are working on a different genus very far away from Pristionchus, where you do not have 2 different morphs, you have 3 different morphs here. All 3 morphs are predators. They are predators already in larvae, whereas here it's only the adults. So nature can play around with this like crazy. And finally, canalization, like we have seen it in the lab, has also occurred in evolution because they are highly derived species, genera, where you have no teeth anymore or where you have no plasticity. So you see that all of this is important for evolution. And when we applied Bayesian analysis on these morphometric traits, then it turned out that the origin of plasticity and the phenotypic fixation are important for the evolutionary diversification. So I have already mentioned Atta and Sheila. The rest of the lab is, of course, similarly important. And I should use this opportunity also to thank my former postdocs that now run their labs around the world, because without this community effort, we would not have been able to get that far. Thank you very much.
So according to have your model of the assimilation, like there would be like a macroevolutionary tendency to lose plasticity in some way, no? Or how likely do you think you can evolve plasticity back once you lose it?
So you can of course independently gain again, but alone in our genus Pristionchus, we have by now 51 species, and we see that in the genus multiple species. So this canalization, I think, is and we also have, I should say, we have Pristionchus pacificus wild isolates that are under laboratory conditions completely colored. So no matter what you do to them, you cannot get them to have the home predator. One issue that is of course very important is that you can afford to be predatory only if you are a mother. Because there is of course the element of cannibalism. So Pristionchus pacificus can distinguish kin from non-kin. So if you have two different strains of the same species, then they will kill each other. And the majority of Pristionchus species, the majority of nematodes are of course monogamous, male and female. That means you always mate with a stranger that is genetically of a different genotype, and therefore all of these monogamous species are preferentially non-predatory because you have trade. Do you want to mate or do you want to kill your kids? And we have been able to show that if you take these male and female species and force them to have the predatory mouse form, kids will kill their parents and parents will kill them. So there's this additional element. Yeah.
So this is a fascinating study. I was wondering about the microRNA. Is it transmitted from the mother to the progeny? Is this period of the persistence in the progeny important? And also, can you force overexpression of microRNA? What happens?
So,
What is
yeah.
the result?
So the microRNA is involved and it's crucial for memory. The ubiquitin ligase comes in to destroy your ubiquitinase alveolate, and then the microRNAs are degraded. As long as the ubiquitin ligase is not active enough, there is no memory. And the memory only kicks in when the ubiquitin ligase will destroy the microRNAs. We have, it's a horrible story. So there is a 10 kb area with 44 identical copies of the same microRNA. And we had to eliminate the complete cluster. And if we do this, we get memory early and we get memory extended, which clearly shows that the microRNA inhibits memory to occur in the first place.
Overexpress the microRNA?
We are still in the process of trying this, but knocking out is easier in our system. And for whatever reason, in C. elegans, you can put in a 10 kb piece of DNA and implant the genome. In Pristionchus, we are limited to a couple of hundred base pairs. We still don't know what the reason for that stuff is.
So does that mean that canalized predatory phenotype is kind of an evolutionary dead end then, if it's in post-hermaphroditism?
You could call it this way. I wouldn't call it this way. They can regain plasticity from another source. But with respect to this particular evolutionary path, this is
Is that what? Sorry, I was trying to ask you, like, have you ever tried to identify like the specific chromatin accessible region? Like,
yes, that is what we are doing, and it's all chromatin manipulation. And I think also for this audience, the next level of understanding is that. So in nematodes, we don't have DNA methylation, so we are one one problem short of other systems, but we have, of course, many histone modifications, acetylation, methylation, and there are 50 enzymes for giving these marks or removing these marks. And what we are as a community, what we have in general biology, what we have to think of is that there are the overwhelming majority of RNA modifications are not novelties. We know now the modifications at histones, and we know many of the enzymes, but organic chemists believe that mRNAs are modified by at least 120 different types of variants, and how that will play into noise and stochasticity, nobody knows. So therefore, when we yesterday discussed the difference in physics, in biology, we still have to work on our understanding of molecular dynamics of life. We do not understand anything that is out there. So biologists are happy with the molecular dogma, DNA makes RNA makes protein. The organic chemist says, no, we don't understand RNAs, mRNAs at all. There are so many modifications, and all of this will be important in some.
Perfect. Thank you very much, Ralf.